Welcome guys, welcome to the another video. In this video we will be talking about the Gfr and factors affecting on Gfr. So stay with me and know about this topic. First of all, we will try to understand the basic things. Then we will go in detail about the Gfr and factors affecting on Gfr. As we know, our heart which pumps the blood, and by the help of a renal artery our kidneys receive the blood. So let me draw here, then we will get an idea about the kidneys. Suppose this is the heart. It's not a professional diagram, just make it only for the understanding purpose, then you will get an idea. Suppose this is the kidney and this is the heart. The job of the heart is pump the blood. By the help of a renal artery, our kidneys receive the blood. Okay, inside the kidney there are two portions. Inside the kidneys there are two portions. The first one is cortex and the next one is medulla. So inside the kidney there are two portions, or there are two regions. The first one is cortex region and the next one is a medulla region. Inside the kidney there are present so many nephrons. Okay, the functional unit of kidney is a nephron, which is actually filtrating the blood and produce the waste material or produce the urine. So inside the kidney there are present in one kidney there are present 1 to 1.2 nephrons, and in both kidneys there are present 2 to 2.4 nephrons. So in two kidneys there are present 2 to 2.4 nephrons. By the help of nephrons, our kidneys produce the waste material, our kidneys produce the urine. Now we will try to understand the nephron. Okay, how the urine is actually formed, or what type of things present in a nephron. So first of all, let me clear this. This is the efferent arteriole. Okay, this is the efferent arteriole, and the efferent arteriole quickly divides into many capillaries. This is the efferent. So this is the efferent. Okay, this is the efferent. This is the efferent arteriole, and this is the efferent arteriole. Remember one thing: the efferent arteriole diameter is more than the efferent arteriole. So the diameter of the efferent arteriole is more than this efferent arteriole. Remember this. The efferent arteriole divides into many capillaries, many capillaries. These capillaries are fenestrated capillaries. I will tell about what is the fenestrated. So these capillaries are surrounded some cells. Okay, these are the cells. These are the cells. These cells are called visceral epithelial cells. Okay, let me recap once again. This is the efferent arteriole. Okay, the efferent arteriole divides into many capillaries and finally rejoins again and made the efferent arteriole. And these capillaries are surrounded some cells. These cells are called visceral epithelial cells. And here are the another package of cells. Here are presented another package of cells, and these cells are called parietal epithelial cells. So what we done? These are looking very happy. So these capillaries are surrounded some cells, some layer of cells. There are present two layers. The first layer is this visceral epithelial cells, and here is another layer that's called the parietal epithelial cells. And this is the another portion is called PCT, proximal cannulated tubule. Now what happened here? As I said, the capillaries are fenestrated capillaries. Fenestrated means there are present tiny holes. In capillaries there are present tiny holes. Okay? There are present tiny holes, very small holes. And here are present another package of cells. Let me clear it first. So these are the fenestrated capillaries. The fenestrated capillaries are the capillaries have many holes, small tiny holes. The diameter of those holes are 70 to 100 nanometer. Okay, the capillaries are fenestrated capillaries, and the fenestrated means there are present small tiny holes in capillaries, and the diameter of those holes are 70 to 100 nanometer. Okay, here are the another package of cells. Here, these cells are called mesangial cells. What is the function? I will tell you about the function of the mesangial cells. So it was just the overview. Okay, now here are also the here are sheet-like. Here are another layer or sheet-like structure that's called GBM, glomerular basement membrane. Now we will enlarge only. We will take from this structure only this part, and we enlarge it. Then we will try to understand how the filtration occurs here. Okay? Let me try here. So this is the afferent arteriole. This is the afferent arteriole. This is the afferent and this is the efferent. Afferent. This is the GBM, glomerular basement membrane. Okay, and here are present mesangial cells. There are also present mesangial cells. We have present capillaries. These are so many capillaries here. Okay, and the capillaries are fenestrated capillaries. Means they are small tiny holes. By the help of these holes, the filtration occurs here, and the diameter of these holes are 70 to 100 nanometer. Okay, and this is the another sheet. This is the GBM, glomerular basement membrane. after this glomerular basement membrane, there are present some cells. There are present some cells. So these cells are looking very happy. These cells are called visceral epithelial cells. Visceral epithelial cells. Okay. Here are the another package of cells. Same in this side. Capacity. Same in this side, there are present also the cells, and these cells are called parietal epithelial cells. Parietal epithelial cells. Same in this side, but I only enlarge it. Okay. So this is the afferent arteriole. The afferent arteriole, here are the a bunch of capillaries that's called the glomerulus. Glomerulus, it's nothing, it's a bunch of capillaries, or it's the ball of capillaries. Okay, the capillaries are fenestrated capillaries, means there are present some small tiny holes. By the help of tiny holes, the filtration occurs here, and these capillaries rejoin here again and made the efferent arteriole. So this is the inlet. Okay, this is the inlet, and this is the outlet where the blood leaves finally. So the inlet where the blood comes, okay, and here the filtration occurs. Here are present so many capillaries, the bunch of capillaries that's called the glomerulus. Now come here. This is the another sheet here are present. This sheet is called GBM, glomerular basement membrane. Okay, after the glomerular basement membrane, here are present cells. Okay, these cells are called visceral epithelial cells. Okay, the visceral epithelial cells, there are present parietal epithelial cells. These cells are made the Bowman's capsule. Okay, now we will call it Bowman's capsule. These cells made the Bowman's capsule. So this is the Bowman's capsule. This is called the Bowman's capsule. Now what happens here? When the blood comes, here are the filtration occurs. The GBM is in middle. It's looking very dense under the electronic microscope. When we see this GBM under the microscope, under the electronic microscope, it's looking in between, looking very dense. So it's called lamina densa. And this side, the upper side, which is facing towards these capillary body or endothelial cells, which are facing towards the capillary cells. This part is called lamina rara interna. So the middle part of this GBM is called lamina densa, and the upper part of this GBM is called lamina rara interna, and the lower part, this part, okay, try this lower part, it's called lamina rara externa. So the GBM has also three regions. We can say it has also three regions. The first one is lamina densa, which is present in between. By the help of electronic microscope, we can see this. In between, the GBM is looking very dense. That's called the lamina densa, and the upper part of this GBM is called lamina rara interna, and the down part of or the below part of this GBM is called lamina rara externa. After this GBM, here are present the cells. Okay, as I said, this is called visceral epithelial cells. These visceral epithelial cells, they are interdigitate with each other. They are looking like this, they are interdigitate with each other. Okay, these cells, these visceral epithelial cells are connected with each other. Okay, connected with each other. Here are present filtration slits. Okay, are like this. Let me. Like this, they are like interdigitating each other. Okay, interdigitating each other. And here are present some space like this. Here are present some space. This space is, here are present filtrating slits. Okay, filtrating slits. Here are present filtrating slits, and the diameter of the filtrating slits are 20 to 30 nanomicron. Okay, the filtrating slits diameter is 20 to 30 nanometer diameter. Here are present, it's recently known, some scientists found here are present the protein. Okay, here are present scientists found here. There are present the protein. That protein is called, that's the special protein, that's called nephrin. Nephrin. Okay, so here are present the protein that's called the nephrin, and here are present the filtrating slits. So when the blood comes here, okay, when the blood comes here, the blood is passing from this these capillaries to GBM. After the GBM to these visceral epithelial cells, finally goes down. Okay, but those only those things can filtrate which have 70 to 100 nanometer diameter, so they can pass easily from this portion to this portion. But those particles can further filtrate here, those have 20 to 30 nanometer diameter. So here are some cells, RBCs, WBCs, platelets. They cannot pass this space and comes in this side, because the RBCs or the WBCs have the more size than this. So the RBC, WBC or platelets, they cannot pass this these capillaries and comes in Bowman's capsule, because the size of RBC is about 8 to 10 micron. Okay, and the WBC size is about 12 to 14 micron. So they cannot pass this membrane and comes in this side and goes to the urine. Or platelet about 1 to 2 micron. Okay, but here is question arises. Okay, here is the question arises. Here is the plasma protein. Some plasma proteins like albumin, immunoglobulin, and fibrinogen. The albumin protein, the albumin protein is the albumin protein can pass this membrane easily, or the albumin membrane has enough size for filtration. So why the albumin cannot pass this side and comes in this side and goes to the urine? Because the GBM and the visceral epithelial cells, or these capillary endothelial cells, are negatively charged. Okay, they are present in negatively charged. And the albumin is also the negative charge. So when the albumin comes here, so here are repelling with each other, the albumin goes back. That's why the albumin does not filtrate here. So the albumin has the right side, but the albumin has the right side to filtrate this. The glomerulus can filtrate the albumin and goes to the urine. But the albumin is the negative charge, and the GBM is also called the negative charge, and the visceral epithelial are also negative charge, and the endothelial cells are also here are also present the negative charge. That's why the albumin is not filtrating. Now I hope you understand this glomerular, this nephron. Now we will be talking about the factors. What type of factors, or what kind of factors which are affecting the GB and the GFR. Here are some factors like hydrostatic pressure. Okay, the hydrostatic pressure, the osmotic pressure, the glomerular filtration pressure, also the efferent concentric, these efferent can block, that's also the another factor. So let me first of all clear it. The first one is hydrostatic pressure. Hydrostatic pressure. The next one is osmotic pressure. There also the another glomerular capillary pressure. Also the present consent of afferent arteriole, and also the afferent arteriole. These are some factors. The first one is hydrostatic pressure. Okay, the hydrostatic pressure. When the hydrostatic pressure increases, okay, it's caused due to the some obstruction or kidney edema. Okay, when the hydrostatic pressure increases, the GFR also decreases. Okay, so when the hydrostatic pressure increases, the GFR decreases. The hydrostatic pressure is about 15 mm mercury. The normal hydrostatic pressure is about 15 mm mercury. And the next one is osmotic pressure, which is exerted by the plasma proteins. Okay, the osmotic which is exerted by the plasma proteins. When this osmotic pressure increases, the GFR decreases or the GFR reduces. The normal osmotic pressure is about 25 mm Hg mercury. And then the one is glomerular capillary pressure. The glomerular capillary pressure, when it increases, the GFR also increases. It's about 25 mm mercury. Okay, the first one is hydrostatic pressure, the next one is osmotic pressure, and the third one is glomerular capillary pressure. Here are also the characteristic of afferent arterioles. For kind some obstruction occurred here in afferent arteriole, the GFR also decreases or reduces. Same in outlet in afferent, same in afferent arteriole. Here happens something, so the GFR decreases or GFR reduces. So these are some factors which can affect the GFR. I hope you like this video. If you like this video, share this video, comment this video. Thank you.